So friend, likewise we have seen that the enzymes are having high specificity. Why? Because in the active site of the enzyme there will be presence of specific amino acid. म्हणजे दोन एन्झाईम जर असतील तर दे कॅन कॅरी आउट द स्पेसिफिक बंच ऑफ रिॲक्शन. ह्याचे उदाहरण देत असताना मी तुम्हाला डायजेस्टिव्ह सिस्टीमचे उदाहरण दिले, सलायव्हरी ग्लँडच्या सलायव्हरी अमायलेजचं उदाहरण दिलं, पॅनक्रियाटिक डायजेस्टिव्ह एन्झाईमचं उदाहरण दिलं होतं. आणि हे शिकत असताना आय हॅव टोल्ड यू मेनी मोर टाइम दॅट की one enzyme carries some reactions that cannot carry other enzymes reactions. So because of the specificity of that particular enzyme, and with this specificity and features, अरे हा कोण आहे, हा हे करतो आहे, with this specificity
मग जास्त
हॅलो
2 एकर आहे ना रे.
अरे तन्मय काय बोलताय बेटा?
हो सर.
बोलताय का तुम्ही? म्यूट करा तुमचे माइक. So mechanism of enzyme action. So in enzyme catalyzed reaction, three components are present. That is the substrate, will be there. Then you can see the enzyme and product. So later on the product will form where first of all substrate and enzyme complex will form, and then enzyme, this substrate bind to the active site of the enzyme, and then ultimately product will form and enzyme come out from the reaction as it is. So when enzyme substrate unite or mix, some reaction takes place and product form. So when enzyme substrate form, at the time enzyme acts on substrate and convert it into the product and remain as it is. So the enzymatic reaction can be represented as, the enzyme plus substrate so come together to form the enzyme substrate complex. So such a way this enzyme catalyze the reaction, and within fraction of second it convert this substrate into the product. So then enzyme will release from the reaction as it is, so it can catalyze other reactions, similar reactions, and then we will form the product. So this is the mechanism of action of enzyme. The reaction rate can be determined by either measuring the rate at which substrate is used or the rate at which product is formed. So friend, how we can calculate or we can count the rate of reaction? So we can simply calculate how much substrate is utilizing or used in the reaction, or the other way is how much product is forming in a reaction, in particular reaction. So this is the way the rate of reaction can be determined or calculated. So the enzyme activity expressed in international unit, that is micromole substrate consumed. So whatever the substrate reaction have been catalyzed, so we can there are two international ways to count the activity of enzyme, that is how much substrate is consuming in the reaction, that is micromole. You can see here that is micromole of substrate consumed in the reaction, or other way is micromoles of product formed in 1 minute under the optimum condition of temperature and pH. So friends, what is optimum condition? मी तुम्हाला सांगितलं होतं की प्रत्येक एन्झाईमची ऍक्टिव्ह असण्याची ऑप्टिमम कंडिशन्स ह्या दे आर स्लाइटली डिफरंट. हे शिकत असताना आय हॅव गिवन यू एक्झाम्पल ऑफ पेप्सिन. Pepsin is the enzyme which is present in the stomach and which digests the protein. So we know that what is the pH of stomach? That is 1.5 to 2. So this is very much acidic pH. Okay. So in such acidic pH, this enzyme get activated. And on the other hand, आपण दुसऱ्या बाजूला पाहिलं होतं की there will be pancreatic amylase, pancreatic lipase. These enzymes, pancreatic enzymes, are active in alkaline environment, or simply we can say 7.5 to 8 pH environment. So this is the way, these are the optimum conditions. Means pepsin is active in the acidic pH, whereas the pancreatic amylase and lipase is active in the alkaline pH. So these are the optimum conditions for different enzymes. So here we should notice that the optimum conditions of every enzyme, they are slightly variable, थोड्या वेगवेगळ्या असतात. So such a way. So friend, what is enzyme substrate complex? So according to the most of the hypothesis, the enzyme combines with the substrate to form the enzyme substrate complex, which get converted into product and enzyme. So simply enzyme and substrate come together. Generally enzyme is slightly larger as compared to substrate. Then the enzyme substrate complex will form, and this enzyme catalyze the reaction and substrate get converted into the product. So then again enzyme combines with other substrate to form enzyme substrate complex. So in every reaction we know that the enzyme cannot get digested, or enzyme cannot convert into other molecules. So enzyme will be released as it is in every reaction. So it can catalyze other similar reactions. Okay. So the substrate attached at a specific point on the enzyme, and this point of attachment is called active site. So friend, I have shown you a lot of time that enzyme is globular protein. So enzyme is globular protein. It is having the active site. So this is the active site of this enzyme. You can see. So here the suitable substrate. Here suppose this is substrate. This suitable substrate will come and bind at the active site of that particular enzyme, and this particular bunch of amino acid make the active site of the enzyme. They are called as active site of that enzyme. So according to Emil Fischer, lock and key hypothesis, substrate molecule fixed into the active site of the enzyme as a key into lock. So the scientist known as Emil Fischer compare this enzyme substrate complex with lock and key. So he just simply compare that the enzyme substrate come together just like lock and key. So concept of active site. So concept of active site is that as the substrate molecule are comparatively very much smaller, so substrate molecules are very much smaller than the enzyme molecules. That means the enzyme molecules are larger. There should be some specific sites on the enzymes. There should be some specific sites on the enzyme for binding with substrate. So friends, this globular protein, I have told you that enzymes are globular protein. So they must have some specific sites. So in these sites, in this such sites, substrate can get fixed. So otherwise, if the substrate and enzyme, so enzyme's active site and substrate does not get matched, they cannot get fit into each other, and enzyme cannot properly catalyze the reaction. So enzymes should have specific sites for binding the substrate. So such site of attachment variously called as active site. So see, such sites are called as the active sites or substrate site. Substrate site. So where the substrate is coming inside the enzyme and get fixed get fixed into the enzyme, and then enzyme can catalyze the reaction. So this diagram, friends, see this diagram. So this is the active site. Here you can see how this active site have been shown here. So this is the active site of the enzyme. So in this active site, you can see the zigzag shape of this active site of enzyme, and then on the upper side greenish in color, that is the substrate. So you can see the zigzag same shape of the substrate. So the enzyme and substrate get come together very closely. So the enzyme can catalyze or convert the substrate into the product. So this is the concept of active site. So this is the thing, friends, concept of active site. So active site possesses some common features. So what are the features active site is having? So there are some few features active site is having. The active site occupies a relatively small portion of enzyme. So enzyme molecule, the active site occupies a relatively small portion of enzyme molecule. Means, see simply, this is the whole enzyme. This is very much larger. But see, you can see here, this is the active site, friends. This is the active site, and this whole globular structure is supportive structure of this enzyme. Okay, but active site only this. So the active site occupies relatively small area or portion. So the active site is three dimensional and made up of groups come from different part of amino acid sequence. So friends, we know that basic structure of amino acid is made of carboxylic group, amino group, and hydroxyl group. So these different groups combination form the different amino acid, and there will be, friends, 2021 significant amino acids are there. So different amino acid arrangement make the different active site of various enzymes. Okay, so this active site make the three-dimensional structure from different to form the different enzymes. Okay, so usually, अरे हे कोण काढतंय यारो, please stop this. So usually arrangement of atom in the active site is well defined, resulting the marked specificity of enzyme. So usually arrangement of atom in the active site मध्ये अमिनो ऍसिड अशा पद्धतीने अरेंज केलेल्या जातात, तर वेगवेगळ्या एन्झाईमची वेगवेगळी ही अरेंजमेंट असते. अँड बिकॉज ऑफ this provide the specificity of the enzyme. The active site bind to the substrate by relatively weak bond. So this is very much important thing, friends. You should remember that the substrate binds to the enzyme by weak forces. Weak forces means the bond is very weak. So because why bond should be weak? So as soon as substrate is get converted into the product, the product must be released very quickly. Product पटकन त्या enzyme मधून बाहेर पडायला पाहिजे, and then again substrate should come into the active site, and that the enzyme should be ready to accept that substrate. Because of this reason, the enzyme substrate complex is bind by weak forces, for example like hydrogen bond or other weaker bonds, other attractive forces. So such a way the enzyme substrate complexes are not bind by covalent bond or any type of strong bond. Okay, so they are bind with enzyme substrate complex bind with weak forces or weak bonds. The active site, next fifth feature of the active site is the active site in the enzyme molecule are groove or crevices from which water is largely excluded. It contain the active site contain different amino acids. See, the active site contain the different amino acids and the side chain group like I have told you carboxyl group COOH, amino group NH2, and then hydroxyl group. Okay, so these groups have been arranged differently. So for what purpose? For providing the specificity for every enzyme. So, and these groups particular present in different arrangements serve as catalytic groups in the active site of the enzyme. So, such a way, these groups which are generally present in the amino acid, that is carboxyl group, amino group, and hydroxyl group, they perform the different catalytic functions or specific functions in different active site of the enzyme. So Fisher lock and key model. So previously, the interaction of substrate and enzyme was visualized in terms of lock and key model. So now just the activity of enzyme is calculated or observed, but observed is different. But previously, the interaction of substrate and enzyme was visualized in terms of lock and key model. So, and this lock and key model proposed by Emil Fischer in 1898. So, according to this model, the union between substrate and enzyme takes place at the active site, more or less in which a key fits into the lock and result in the formation of enzyme substrate complex. So these things I have told you. Then the enzyme substrate complex is highly unstable. See here, the the enzyme substrate complex is highly unstable, and almost immediately this complex decomposes or detached to form the end product of the reaction, and enzyme regenerated. What does it mean? The enzyme get released in the reaction and substrate is converted into the product. So this concept developed to explain high specificity of enzyme. ज्या पद्धतीने प्रत्येक लॉकला वेगळी की असते नाही का, त्याच पद्धतीने या आपले जे सायंटिस्ट आहेत नोन ॲज एमिल फिशर, ही वॉन्टेड टू शो दॅट की एव्हरी एन्झाईम can catalyze different type of reactions. So he compared, because of that, he compared the model of lock and key. So according to this concept, a structurally well-defined catalytic site will accept only those substrate molecules which have a matching shape. See, so in such type of enzymes, only such type of substrate have been accepted which are having the specific or matching shape. So which have matching shape and will repel other different structure or other type of substrate which are having different structure. रिपेल म्हणजे परावर्तित करणे, त्याला ऍडजस्ट न करणे. So this is the way the scientists known as Emil Fischer have shown the lock and key model So next model is after lock and key model proposed by Mr. Emil Fischer. Next model is the Koshland's induced fit model. So Koshland's induced fit model, in order to explain the enzyme properties more efficiently, the scientist known as Koshland in 1958 modified the Emil Fischer's model and presumed that enzyme molecules does not retain its original shape and structure. Means, what does it mean, friends? म्हणजे ज्या वेळेस सबस्ट्रेट हा एन्झाईमच्या ऍक्टिव्ह साईटला जातो त्यावेळेस एन्झाईमच्या ऍक्टिव्ह साईटमध्ये काही विशिष्ट बदल होतात, विशिष्ट चेंजेस होतात, असं आपल्या मॉडेलमधून मॉडिफाईड मॉडेलमधून सायंटिस्ट ज्याचे नाव आहे कोशलंड यांनी दाखवलं की देर विल बी सम चेंजेस आफ्टर कमिंग ऑफ द सबस्ट्रेट इंटू द ऍक्टिव्ह साईट ऑफ एन्झाईम. ज्यावेळेस सबस्ट्रेट हा एन्झाईमच्या ऍक्टिव्ह साईट मध्ये येतो देर विल बी some changes occur in the active site of that enzyme. The contact of the substrate, see here friends, the contact of the substrate induce some configurational, the contact of the substrate ज्यावेळेस सबस्ट्रेट एन्झाईमच्या कॉन्टॅक्ट मध्ये येतो, इंड्यूस म्हणजे स्टिम्युलेट, so that substrate will induce or stimulate that enzyme. आणि स्टिम्युलेशन काय होतं तर कॉन्फिगरेशनल जॉमेट्रिकल चेंजेस, म्हणजे त्या एन्झाईमच्या ऍक्टिव्ह साईट मध्ये देर विल बी सम चेंजेस, स्लाइटली चेंजेस होतात अँड इन द ऍक्टिव्ह साईट ऑफ द एन्झाईम मॉलिक्यूल. सो दिस हॅव बीन proposed by the scientist known as Koshland. So consequently the enzyme molecule is, and changes, why changes must occur at the active site of the enzyme for the proper fitting of the substrate. Consequently the enzyme molecule is made to fit completely. म्हणजे हा जो सबस्ट्रेट मॉलिक्यूल आहे आणि एन्झाईम आहे ते एकमेकांमध्ये कम्प्लिटली फिक्स झाले पाहिजे फॉर प्रॉपर कॅटलायझेशन ऑफ रिॲक्शन. प्रॉपर सबस्ट्रेटचं रिॲक्शन कॅटलाइज करण्यासाठी ते हे चेंजेस खूप गरजेचे आहेत. आणि त्यामुळे कोशलंडनी आपल्या त्या फीचर्स मध्ये सांगितले त्यांच्या मॉडेलच्या की consequently the enzyme molecule is made to fit completely the configuration and active centers of the substrate. So at the same time other amino acid residues may become buried. So at the same time other amino acid residue may become buried in the interior of the molecule. म्हणजे मित्र हो, ज्या वेळेस सबस्ट्रेट हा एन्झाईम मध्ये येणार आणि सबस्ट्रेटच्या ऍक्टिव्ह साईट एन्झाईमच्या ऍक्टिव्ह साईटला देर विल बी सम चेंजेस. सो वाय दिस चेंजेस टू फिक्स द सबस्ट्रेट इन टू द ऍक्टिव्ह साईट ऑफ द एन्झाईम. आणि तसेच एन्झाईमचे काही जे बॉन्ड्स आहेत अमिनो ऍसिडचे बॉन्ड दॅट विल बी गेट कनेक्टेड टू दी सबस्ट्रेट. आणि कशासाठी? फॉर चेंजिंग सबस्ट्रेट प्रॉपरली इन टू दी प्रोडक्ट. So this have been proposed by a scientist known as Koshland. So this hypothesis, see here, so this hypothesis seems attractive because the protein molecules is flexible enough to allow conformational changes, and give some explanation for the influence of hormone on the enzyme activity. म्हणजे काय? आता हे कन्फर्मेशनल चेंजेस मी म्हणजे हे सायंटिस्ट आहेत कोशलंड नावाचे ज्यांनी आपल्याला सांगितलं की एन्झाईमच्या ऍक्टिव्ह साईटला देर विल बी सम चेंजेस आणि ते कोण इंड्यूस करतो? सबस्ट्रेट करतो. तर हे या स्टेटमेंटमध्ये शेवटच्या त्यांनी सांगितलं की इट इज मोस्ट अट्रॅक्टिव्ह. वाय? बिकॉज जर आपण इन्सुलिनचं उदाहरण घेतलं तर वी शुड नो दॅट व्हॉट इज द हार्मोन नोन ॲज इन्सुलिन रिलीज बाय पॅनक्रियाज. पॅनक्रियाज मधून इन्सुलिन हार्मोन रिलीज होतो. आणि व्हॉट इज द फंक्शन ऑफ इन्सुलिन? ग्लुकोजचा अपटेक करण्यामध्ये ह्या हार्मोनचं खूप महत्वाचा रोल आहे आपल्या शरीरामध्ये. इन्सुलिन ज्या लोकांमध्ये कमी प्रमाणात असतो किंवा नसतो त्याला शुगर झाली किंवा डायबिटीज झाला आपण असं म्हणतो, तर हा हार्मोन काय करतो, सेल्स वरती जाऊन बाइंड होतो पर्टिक्युलर साईटला आणि त्या सेल्स काय करतात, ग्लुकोज आतमध्ये घेतात. पर्टिक्युलरली हा हार्मोन सेल्सच्या पर्टिक्युलर प्रोटीनवर जाऊन बाइंड होतो, जो की ज्यामुळे ग्लुकोज हा ब्लड मधून आपल्या प्रत्येक सेल्स मध्ये घेतला जातो आणि कशासाठी, फॉर फॉर्मेशन ऑफ एनर्जी. तर या पद्धतीचे मेकॅनिझम जे आहेत, या पद्धतीचे जे मेकॅनिझम आहेत, this will get fixed into the Coastlands induced fit model. And because of this, after the proposal of Mr. Emil Fischer, which proposed the lock and key model, this was the modified model of enzyme activity. So this is the case, friend. Now we will see so here you can see how the enzyme catalyze the reaction. So here first of all, enzyme substrate come together. This is the enzyme, this is the substrate. So you can see the active site of the enzyme. This is the active site of the enzyme, particular enzyme. This is the active site, and see the shape of the substrate, and then you can see the enzyme substrate complex. So in which the substrate get fit into the active site of the enzyme. And why? Because there will be some changes occur in the active site of the enzyme, and this active site of this enzyme have adjusted this substrate, and then within a fraction of second, friends. So here you can see enzyme changes shape slightly. So this is very much important. So this shows us the Coastlands induced feed model. So here enzyme changes shape slightly as substrate bind. And why? What is the purpose? Because this enzyme wanted to convert this substrate into the product. So here you can see, friends, the enzyme product complex. So you can see the different colors. So there will be formation of product. So there will be formation of product. You can see here, because in this diagram, ह्या डायग्राम मध्ये कलर चेंज करून दाखवलेले आहेत की देयर विल बी सबस्ट्रेट कोणत्या कलर मध्ये बाइंड केला आहे, ग्रीन कलर मध्ये, and there will be formation of product in the coloration of gray and orange. So after formation of enzyme and product, after within fraction of second, the product, see here, the product get released from the enzyme, and see here enzyme remain as it is. Enzyme did not get digested, so products leaving active site of the enzyme. And then now in this step your enzyme is ready to complete again same reactions. So this is the way, friend. These are two very significant Emil Fischer's lock and key model and Coastlands induced fit model. So this is the thing regarding the introduction of enzyme, the properties of enzyme, then characteristic of enzyme, and then what is active site, and then the different examples which and hypothesis proposed by different scientists. So Emil Fischer, he has proposed the lock and key model, then Coastlands induced fit model, he has proposed the better or modified version of the Emil Fischer's lock and key model. So this is regarding the enzymes, friends. So today we will